If you're anything like me, you've probably not given much thought to your breathing and digestive processes until you get short of breath or hungry. I always assumed that I just breathed in oxygen and exhaled carbon dioxide without really understanding how that happened. Similarly, that what I ate just got absorbed into my body and oxygen got transformed into the energy we need to live until I started putting together this presentation on the mitochondria. And the deeper I've gotten into it, the more impressed and awestruck I have become at the remarkable and I believe unmistakable signs of the Creator's design that can be seen in the mitochondria and the processes embedded in them. So fasten your seatbelts and join me on this exploration of the mitochondria. Mitochondria are tiny organelles found in almost every cell of our bodies. In many ways, mitochondria can be thought of as cells within cells. Mitochondria take the glucose in our body and the oxygen we breathe and convert them into energy. Mitochondria exist in almost all living organisms except for bacteria and archaea. And it turns out that even though bacteria and archaea don't have mitochondria, they do share most of the same biochemical processes that are found in mitochondria. I'll focus this talk on the human mitochondria, although for the most part it could apply to nearly any other organism as well. Mitochondria are often called the powerhouse of the cell because they provide energy for most of our bodily processes. They take the oxygen we breathe and break down or oxidize food into something called adenosine triphosphate, or ATP, carbon dioxide, and water. But mitochondria also provide other essential roles, which I'll mention later. The subtitle of this slide and of this presentation is, I've got a protein for that, because proteins are incredibly complex and yet essential. Each one has a clear purpose, and they work together to perform those purposes in complete coordination with each other. So by the end of this presentation, you will have heard me say, I've got a protein for that, quite a few times. Almost every cell in our bodies has mitochondria, and the number varies widely depending on the cell's energy needs. Typically, there are 1 to 2,000 in a cell, but there are a lot more in many cells and quite a few less in some cells. Mitochondria take up a significant percentage of the cell volume, about 20%. And the main purpose of this lecture is to explain what mitochondria are and how they work, and at the same time to provide evidence that mitochondria point to design. And to do that, I will use the watchmaker argument, as well as showing how scientific evidence discovered about mitochondria is best explained by design, not by evolution. And I will argue that multiple processes in mitochondria show clear evidence of purpose and appear to me clearly to be designed. Here's an artist's conception of a mitochondrion. In the next few slides, I'll be telling you about how mitochondria work and where they come from. For now, please notice that mitochondria have two membranes, an outer membrane, the yellow band colored sort of blue-green inside, inside and out by the artist, and an inner membrane, the white band colored sort of tan inside and out, which, as you can see, has lots of folds, which increase its surface area. So here's the outer membrane and the inner membrane, and then the matrix, which is the inside. The inner membrane surface area determines how much energy the mitochondria provides, and the space between the outer and inner membranes is called the intermembrane space. Duh. And the space inside the inner membrane is called the matrix. That's this blue area with the little red squigglies. The little red squigglies, by the way, are DNA. And the DNA, mitochondria have their own DNA, as well as their own ribosomes, but most of the cell's DNA is actually stored in the cell nucleus, but the mitochondrial DNA is an exception, and we'll talk more about that later. Here's another view showing a tomographic image of a mouse heart mitochondria on the left and the middle, and an artist's rendition more precisely of the infoldings of the inner membrane on the right. Notice here that a distinction is made between the inner boundary membrane and the crista membrane. So the inner boundary membrane is this thing here that's kind of smooth all the way around. And then the cristae are these folds that go into the inside. So it's kind of it is a continuous membrane, but the cristae membrane are the ones that are folded. And that's very important because most of the proteins that are embedded in the inner membrane, where all the work is done, are on the crista membrane, not on the inner membrane itself. And although it looks like the crista membrane is just a continuation of the outer membrane, 75% of the crista membrane consists of proteins of the electron transport chain, which we'll discuss in a few minutes. That is to say, more of the crista membrane is actually proteins, not just a membrane. That's kind of incredible. But the proteins aren't shown in this picture, but we'll have a look at some of them a little later. The outer membrane lets ions and small molecules pass through and has about the same composition as the cytoplasm of its host cell, whereas the inner boundary and crista membranes are nearly impermeable. This allows for the buildup of a substantial electrical gradient between the intermembrane space and the matrix, which we will see is essential for the production of ATP. It is a fact that all your mitochondria come from your mother. This is an artifact of the reproductive process. The human egg is much larger than the sperm, and the function of the sperm is just to carry the male DNA into the egg. Although there are mitochondria wrapped around the tail of the sperm, they only rarely get inserted into the egg. Since most of your mitochondria originate in your mother, scientists can use the differences in mitochondrial DNA to trace the human lineage back to a single woman, affectionately called Mitochondrial Eve, who lived many thousands of years ago. This is an imprecise measurement and has an uncertainty of tens of thousands of years at this point in time, and mitochondrial DNA has only about 17,000 base pairs, whereas nuclear DNA has about 3.2 billion base pairs. So the vast majority of mitochondrial proteins, over 1,000 mitochondrial proteins, originate in the nuclear DNA, and only 13 proteins come from mitochondrial DNA. Coordinating the manufacture of proteins from two different sources is challenging, and I'll have more to say about this later. The accepted origin of mitochondria, according to most evolutionary biologists, is called the endosymbiotic hypothesis. The endosymbiotic hypothesis for the origin of mitochondria suggests that mitochondria are descended from specialized bacteria that somehow survived endocytosis by another species of prokaryote or some other cell type and became incorporated into the cytoplasm. The idea is that sometime in the very distant past, most likely when only single-celled organisms existed, a bacterial cell or possibly a very early single-celled eukaryotic organism absorbed another bacterial cell which had the properties of mitochondria, without digesting or destroying it, and the two species developed a symbiotic relationship whereby both benefited from the existence of the other. Over the course of many, many generations, mitochondria are supposed to have evolved to their present form, including losing most mitochondrial DNA to the cell's nucleus. Although this theory is widely accepted among evolutionary biologists, I have significant doubts about it, and many scientists do as well. Here's a quote from a recent research article. The precise bacterial origin of mitochondria and consequently the ancestry of the aerobic metabolism of our cells remain controversial despite the vast genomic information that is now available, according to this article in Science Advances in 2023. I don't want to make fun of the endosymbiotic hypothesis since it does hold sway with a large majority of evolutionary biologists. Nevertheless, there are some obvious problems with the hypothesis, three of which I've listed here. First, endocytosis, sometimes called phagocytosis, is generally the way single-celled organisms eat their prey. The outer membrane essentially engulfs or wraps around the prey, and it is absorbed into the host and digested. There's no known process by which in just one generation an endocytosis event could occur and a symbiotic relationship develop between the two organisms. This would normally take multiple generations and would require the first organism not to digest the second, which would be the usual outcome. Second, over some period of time, the two organisms would have to adapt to each other symbiotically so that each gains some advantage. And I've not seen an acceptable explanation for how this could happen for mitochondria. Finally, mitochondria, as I've said, only have 17,000 DNA base pairs, whereas the smallest known bacterial cell has several 100,000 base pairs, and most bacteria have at least 1,000,000. 17,000 base pairs is only about 3% or less of the number required to fully support the mitochondria, so most of the mitochondrial DNA functions actually occur in the nuclear DNA. This is usually explained as the effect of evolutionary processes over 1 billion or more years, where the overwhelming majority of the mitochondrial DNA functions have become incorporated into the cell's nuclear DNA, leaving only a few basic functions to be performed by the mitochondrial DNA itself. I don't find this evolutionary explanation to be satisfying. Here's an illustration of this paradox. At the top of the diagram is the nucleus, and this is a little bit of mRNA, which they're calling DNA, but it's actually mRNA, and it goes out of the nuclear pore and into the ribosome, which is here. This is a cytoplasmic ribosome, and then 1,200 to 1,600 different nuclear encoded mitochondrial proteins result from this action here, and they're transported through the TOM protein complex, through the TIM protein complex into the mitochondria. Meanwhile, the mitochondria uses its own DNA, that's this red circular thing here, and its own ribosomes to create its own RNA, and they kind of come together basically on the cristae membrane of the mitochondria. Recall that there are up to 1,000 or more mitochondria in each cell that must receive proteins manufactured by nuclear DNA, and the problem of coordination of the mitochondrial protein needs between the nuclear and mitochondrial DNA has got to be daunting. And I've not seen much discussion of it in the literature. For instance, imagine a cell with 100 mitochondria, each in various different stages of needing nuclear proteins. How does the nucleus know how many of which of the nuclear proteins to send to each mitochondria without resulting in too many, too few, or the wrong combination somewhere? And what would it actually mean for the nucleus to know anything, since it doesn't have a brain? Here's a simplified explanation of what mitochondria do. They oxidize molecules of glucose, a very common form of sugar, to form a molecule called ATP, or adenosine triphosphate, which provides almost all the energy used by humans and most other organisms. Glucose is the main end product of digestion, which takes place mostly in the stomach and small intestine. Glucose is transported to mitochondria in the bloodstream, as is the oxygen we breathe. There's actually no fire in the burning process. It is a very highly controlled and efficient process. If you know anything about chemistry, you may be objecting about now that the molecules on the left, these here, don't look anything at all like the ones on the right. I'll get into why this is in a moment. ATP, or adenosine triphosphate, that's that's this guy here, has three phosphate molecules. That's these three molecules here. And it gives up energy when one of those phosphate molecules is stripped off the ATP molecule. And then ATP becomes ADP, or adenosine diphosphate, instead of triphosphate. Some facts about adenosine triphosphate. Typically, the human body stores only about 4 ounces of ATP at one time. Every day, however, the body manufactures and uses well over 100 pounds of ATP. Yes, you read that right. Every day, your body produces and uses up about half your body weight, or, if you're like Karen, it's almost your entire body weight, or more in ATP, even though at any one time only 4 ounces are stored. All plants and animals depend on mitochondria to satisfy their need for ATP. Without mitochondria, they and we could not exist. It is no exaggeration to say that we could not exist without mitochondria. The next part of this presentation may seem a little technical, not too much, I hope. The details are not that important, but I want you to be aware that the details of what the mitochondria do are actually pretty complex. Here's a typical animal or eukaryotic cell showing the nucleus, and that's the purple thing here at the top, which contains the nuclear DNA, and in the lower left of the picture you see these two bean-shaped kind of orange things that are mitochondria. But remember that an average cell will have 1 to 2,000 mitochondria, not just 2, as shown here. The mitochondria are floating around in the cytosol, also called the cytoplasm, the liquid inside part of the cell. And it's in the cytoplasm that the first of 4 different processes leading up to the ultimate production of ATP and carbon dioxide takes place. The first of the 4 processes is called glycolysis. As I said, it actually takes place outside the mitochondria in the cytoplasm, but its main product, called pyruvate, initiates many of the mitochondrial processes. This is a flow diagram of the glycolysis process. I know this looks complicated, and it is, but there are just a few things I want you to get out of the slide. A molecule of glucose, like up here, is transformed in this 9-step, 1, 2, 3, 4, 5, 6, 7, 8, 9 steps process into a pyruvate molecule with a net gain of 2 ATPs. First of all, costs 4 ATPs. 1, 2, oh, no, I guess it doesn't. It only costs 2 ATPs, but then 2 more ATPs are made here and another 2 here. So you actually get 2 extra ATP molecules. Those are the energy molecules I spoke of earlier. Notice that during this process, 2 NADH molecules, and let's see, where's the NADH? Here is the NADH molecules are also byproducts. And NADH has 2 free electrons, which will become important during the final process, which we'll discuss, which is known as the electron transport chain. The pyruvate molecule is not really important either, except it is the input molecule to the next process. There's no particular basis for showing this process in this convoluted shape, moving in a clockwise direction like this, bending down and to the right and then up and to the left, other than as a space-saving artifact. Again, the important facts. Glucose is transformed into pyruvate, 2 ATP molecules plus 2 NADH are gained, and it's interesting that glycolysis takes place in the cytoplasm outside the mitochondria, and if the mitochondria did not exist, glycolysis would be the only energy-producing process in the body, but it would be insufficient to provide all the body's energy needs. Thus, mitochondria are essential for our existence. And to emphasize my argument concerning purpose, here's what the actual molecules involved in glycolysis look like. Glucose here is a 6-carbon sugar, and glycolysis breaks it down into a 3-carbon sugar at this point here. Is that right? No, that isn't right. Sorry. This is a 1, 2, 3, 4 carbon sugar, but it's going to get broken down again to a 3-carbon sugar here. And then ultimately, pyruvate is itself another 3-carbon sugar. And along the way, 10 enzymes indicated by the numbered circles, that's this one, hexokinase, phosphoglucose isomerase, phosphofructokinase, etc. All these with the little blue dots, numbered dots, are enzymes, and they're very complex molecules. They're proteins consisting of strings of amino acids, generally several hundred amino acids long, and made by ribosomes from genes. All 10 of those enzymes are unique, and each one is needed in the exact order shown to get from glucose to pyruvate. For each step of the process, I like to say, I've got a protein for that. Or in this case, I've got 10 proteins for that. I think this is a pretty elegant, planned, and purposeful process. Here's a ribbon diagram of one of those enzymes called hexokinase. This is the first one. And human hexokinase contains over 900 amino acids. By the way, hexokinase, like most enzymes, contains a metal ion. In the case of hexokinase, the metal is magnesium, a catalyst which accelerates the conversion of glucose to glucose 6-phosphate. Other enzymes might include different metals such as iron or copper. It turns out that ribosomes don't put metal ions in proteins. Metal ions are added to proteins by other enzymes during protein post-processing. I'll spare you pictures of the other 9 enzymes, but this one is pretty representative. Okay, that was process number 1. Process number 2 is not really named, and most people don't kind of ignore it, but it's a little short process that takes you outside from the cytosol into the matrix of the mitochondria. Through the mitochondrial intermembrane space and into the matrix. In this process, the pyruvate loses a molecule of carbon dioxide here, and the acetyl coenzyme A molecule, which is produced, will start the third process called the citric acid cycle. Again, notice that a molecule of carbon dioxide here and another molecule of NADH are byproducts of this process. So the third process, the citric acid cycle, takes place in the matrix of the mitochondria that is inside the inner membrane. The acetyl coenzyme A molecule up here has 2 carbon atoms. And when I first saw and heard this process explained in the biology course I took in 2009 at Foothill College, I was awestruck. And I wondered how such a process could have evolved. I still wonder. The cycle starts at the top with acetyl coenzyme A, and in this process, 2 more molecules of carbon dioxide are given off, as well as a molecule of GTP, or guanosine triphosphate, which is another energy-carrying molecule roughly equivalent to ATP. Again, the details are not important, but notice that 3 molecules of NADH, 1, 2, and 3, and 1 of FADH2 down here, are byproducts. And to summarize, the citric acid cycle produces a molecule of GTP, 3 molecules of NADH, 1 of FADH2, with 2 molecules of carbon dioxide, one here, and let's see, and then can't find the other one. Here's the other one, yeah, as byproducts. And every step in this cycle has purpose, and to me, they all appear designed. And just like glycolysis, every step requires an enzyme, even though they aren't shown here. So I've got 10 proteins for the citric acid cycle too. And like glycolysis, most of the enzymes also have metal catalysts placed there by even more proteins. And like glycolysis, this seems to me to be elegant, purposeful, and designed. The final process takes place mostly in the inner mitochondrial membrane, more precisely in the crista membrane. Here's the view of the mitochondria showing the distinction between the inner boundary membrane, kind of the smooth thing around here, and the crista membrane, all these infoldings. And the electron transport proteins are actually embedded in the crista membrane. Like the citric acid cycle, I think the electron transport chain also clearly looks designed, and in my opinion, defies an evolutionary explanation. The essence of this process starts with the flow of electrons from the NADH down here and the FADH2 molecules in the lower left-hand corner, which we saw formed earlier through several proteins. Remember, I've got a protein for that, embedded in the inner mitochondrial membrane. The negatively charged electrons pump positively charged hydrogen ions. So the hydrogen ions are these H plus here, and they're called protons. That's what they really are, is protons. And the negatively charged electrons, which are here going here through the 3 proteins, are the things that pump the protons through the proteins into the space, the intermembrane space. The resulting excess positive hydrogen ions in the intermembrane space flow through the ATP synthase motor. This is the ATP synthase motor here. And it's another protein, and it's embedded in the protein at the right, resulting in the production of many ATP molecules. And this flow of positive hydrogen ions through the ATP synthase motor is essentially an electrical current powering the motor. The final step, where an oxygen molecule combines with 2 hydrogen ions to form a molecule of water, is shown just to the left of the ATP synthase. It's right here. So you get oxygen and hydrogen protons combining to form molecules of water. And here's another look at the proteins in the electron transport chain, this time showing the metal ions through which the electrons pass. The blue line is the path of the electrons. So the electrons kind of go this way, and they end up here forming the way that the oxygen is combined with the protons to form water. So the protons essentially, or the electrons essentially, jump from iron sulfur clusters here to iron sulfur cluster, and then to the Q molecule, and then to a heme molecule. That's this little diamond here, and another diamond, and another diamond, and now a copper atom, more heme molecules before they are present to combine the oxygen and proton to form water. Recall that I said that genes don't specify the metals which most proteins contain. and both the iron sulfur clusters and the hemes are complex molecules manufactured and assembled in these proteins in the mitochondria by still more proteins. Remember, I've got a protein for that. The arrangement of these metals appears to be finely tuned to the energy levels which drive the electron transport chain. Again, I think the whole process cries out design. Here are the two iron sulfur clusters found in the electron transport chain. In this diagram, the yellow atoms are sulfur and the green are iron. And here's a listing of some of the proteins that are needed to form the iron sulfur clusters in bacteria here and yeast and humans. So there's a whole bunch of them required just to make these iron sulfur clusters. And they can't be too far apart or too close in the electron transport chain. They're precisely not randomly placed. If they're too far apart, the electron transport process would break down, and if they're too close, more clusters would be needed. Have I mentioned I have a protein for that? I think I have. Electrons in the electron transport chain also traverse four hemes. Here's a picture of a heme. These are not proteins, but as you can see, they're pretty complex anyway. Hemes may be more familiar to you from their role in hemoglobin in red blood cells. In that role, they're also very important because it's hemoglobin that picks up oxygen in our lungs and carries it in our bloodstream into the mitochondria, where it completes the oxygenation of glucose at the end of the electron transport chain. However, individual hemes must be assembled and placed in the appropriate place in the electron transport proteins by a host of other proteins. Heme assembly begins in the mitochondria, believe it or not, represented in this cartoon here as the yellow space with the E-shaped figure. The process moves into the cytoplasm outside the mitochondria over here for a few steps, then back into the mitochondrion, and heme destined for the electron transport chain then remain in the mitochondria. And those destined to become hemoglobin get sent out here, where they're moved back into the cytoplasm where the globin chains are added. But notice that this process requires 7 enzymes, that is, proteins. Did I mention I've got a protein for that? And here's a summary of 3 of the 4 processes. The first process I described, the glycolysis cycle itself, isn't shown here, but it is the input that provides the pyruvate. So the pyruvate is kind of the key process that glycolysis outputs. The picture shows the outer mitochondrial membrane, that's this outer membrane here, with just 3 of the many infoldings called crystal spaces in the inner membrane, 1, 2, and 3. Yet there are many, many, many infoldings. Most of the mitochondrial action takes place inside the matrix, which is inside the inner matrix, inside the inner membrane, which is the gray area in the middle. And when glucose is plentiful, glycolysis produces pyruvate. But when glucose is not plentiful, there are these 3 other backup paths to produce ATP in the mitochondria. First, amino acids, which are normally the building blocks of proteins, but if there's amino acids available which aren't needed to produce proteins, amino acids can be transformed into acetyl coenzyme A. So can fatty acids, and so can ketone bodies, all be transformed into acetyl coenzyme A, and then they can become part of the citric acid cycle and be turned into ATP. As the diagram shows, all 4 of these substances can be converted into the mitochondria into acetyl coenzyme A, which becomes the input to the citric acid cycle. The citric acid cycle then has 2 main outputs: carbon dioxide, which gets excreted here and heads for the bloodstream and shown exiting the mitochondria on the right. And then it returns to the lungs and you breathe out. And NADH, which then becomes the input to the electron transport chain, shown at the bottom left. And oxygen from the bloodstream then comes in and is transformed into water at the end of the electron transport chain. I particularly like this diagram because it shows the electron transport chain on the wall of a crystal space and two of the ATP synthase motors linked together at the fold of the crystal space. An accurate picture of reality according to recent studies. So unfortunately, the videos that I'm about to show are not going to show that the ATP synthase are actually on the fold of the crystal space. But please realize that they are. And when ATP is created like this, it's called oxidative phosphorylation. I haven't said much about ATP synthase, but I consider it the crowning achievement of this entire process, and it is featured in the two videos we're about to watch. Okay, so the electron transport chain process is explained more fully in the following two video clips. You will see in one of the video clips that the ATP synthase protein is actually a quite complex motor, very much like an electrical motor with a stator, rotor, and bushings. And I believe it's another example of design. One other important point, the oxygen molecule in this process comes from the air you breathe, and it is transported to the mitochondria by the hemoglobin in the bloodstream.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, two mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, two electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of one hydrogen ion for each electron. Next, the two electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. One hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates two full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained. Concentration gradients are a key component of the biological world. The potential energy from these gradients is often used to perform biological work. Here we will focus on hydrogen ion concentration gradients. Hydrogen ions are also known as protons. A gradient exists when there is a higher concentration of a molecule in one compartment compared to a neighboring compartment. This animation will demonstrate how the potential energy that results from a hydrogen ion gradient uses ADP and inorganic phosphate, also known as PI, to synthesize ATP. This process involves an enzyme complex called ATP synthase. Gradients and the potential energy they create are key aspects of the biological world. A good example of the use of a gradient occurs in the mitochondria when ATP is synthesized. ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the two sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. One hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
In 1802, the Reverend William Paley published this book entitled Natural Theology. In it, he put forth this argument. You stumble on a watch in a field. You realize it has not always been there, in contrast with a rock, which very well may have been there forever. You see the watch is intricately designed and elegantly made, you conclude, by an intelligent being. You realize that living creatures also appear intricately designed and elegantly made by an intelligent being, and further, they have the ability to reproduce themselves, a property which even the most beautifully constructed watch does not possess. It is reasonable to conclude then that living creatures are also designed by an intelligent being. Although it was possible even as early as 1802 to see elegance in the form of living creatures, not a lot of details were available, especially at the microscopic level. And many scientists dismissed Paley's argument because they believed humanly designed mechanisms were very different from living creatures. This was true up through the middle of the 20th century. Today, however, biological science has progressed to the point where it is possible to make detailed comparisons between the parts that make up living creatures and humanly designed mechanisms, giving new legs to the watchmaker argument. Paley compared a watch to living creatures. I want to compare a generator to the mitochondria. The generator mixes oxygen with a fuel such as gasoline or diesel, burning the mixture to drive an internal combustion engine and electrical generator to create electrical energy. Mitochondria use oxygen from the lungs and glucose from the digestive system to create chemical energy in the form of adenosine triphosphate, or ATP. The generator is obviously designed for a specific purpose. For me, it's pretty obvious that the mitochondria is similarly designed. Furthermore, the mitochondria reproduces itself within its organism and thus keeps the energy conversion process going throughout the organism's life cycle. Also, the mitochondrial energy conversion process is about 50% efficient, whereas the most efficient gasoline engines are only about 20% efficient, most of the energy being lost as heat. Recent scientific research has revealed even more reasons to believe that mitochondria are designed and not the product of evolution. Here's a blog article written in 2022 by Dr. Fuz Rana, biochemist and president of Reasons to Believe, a science faith organization. This blog article provides links to still more blog articles referencing additional articles on mitochondria that Dr. Rana has authored over the years. All of these blogs are based on Dr. Rana's study of recently published research articles, and they provide further strength to the argument that mitochondria appear designed. If you're interested, you can access this article on the reasons.org website by doing a search for mitochondrial design. I close with two questions and an observation which I've been struggling with and haven't found answered to my satisfaction in the literature. First, how are the thousands of mitochondrial proteins which are manufactured in the cell's nucleus and shipped into hundreds of mitochondria coordinated with the handful of proteins manufactured in each of the mitochondrial nuclei? Imagine the logistics challenge of avoiding shortages or overruns and consequences if they occur. Yet, apparently, they are uncommon. I have a number of books on inorganic biology. Think about that term for a moment. Biology generally involves organic chemistry, but I'm talking about inorganic biology, from which we generally learn that there are a whole host of proteins which have been identified in this process, but for me at least, leave a lot of questions like this only partially answered. We do understand that metals incorporated in enzymes tend to speed up the desired chemical reactions by order of magnitudes. So I'm going to read this anyway. Most mitochondrial and other proteins contain metallic ions such as iron sulfur clusters, hemes, and copper ions. They're not specified by the protein's gene. How is this process driven and controlled? Finally, here's an observation that I find inescapable. These processes which we've just been discussing, glycolysis, the citric acid cycle, the electron transport chain, and especially ATP synthase, the motor that cranks out huge numbers of ATP, are all elegantly, extremely complex, precise, and then they proceed in a highly ordered and sequential cycle, and most importantly, they display purpose and intent. If any one step in the process were to fail, or even to be significantly impaired, the consequences for that organism would be catastrophic. Furthermore, they all appear in almost all organisms in all three domains of life: bacteria, archaea, and eukaryotes. This requires that they date back to the earliest era of biology. This seems to be an insuperable challenge to the theory that they could have resulted from evolutionary processes. My conclusion is that a system this detailed, complex, and purposeful must have been designed by an incredibly intelligent designer. I conclude by the God of the Bible. This concludes my presentation, and I'll be happy to try to answer any of your questions.
I mean, a really wonderful presentation, and I agree that it's a design. But one of the things that you also have to mention is that these proteins can fold in enormous ways.
Right. Good point. Yeah, that's right. I didn't even mention the fact that you got proteins that have to fold after they are created, after the amino acids are assembled by the ribosome. They they go off and they have to go somewhere and be folded into the shape that they need to be folded in the shape that they need to be in, and it's very precise. Um, and so that's one more step that's I didn't mention, but it's Lotar's quite correct that that needs to happen.
My basic understanding of the folding is that it's a function of the actual sequence of amino acids in that protein.
Correct.
Yes.
Correct. And if you look at it more exactly, it basically it's not covalent bonding that happens between the different molecules on the different atoms on the amino acid chain. It's hydrogen bonding between them, and various different kinds of bonding processes that are taking place, and they happen very precisely. And if you didn't have the exact same um, sequence of amino acids, you'd get something entirely different.
Yes. Yes.
So and they're, yeah, very complex, I think.
One of the explanations that I've heard about protein folding is that there is a protein machine, a machine made out of proteins that's only function is to fold proteins in the proper way. And I cannot remember what that particular protein is called.
There are several. So there's not just one. It's a but you're right. There are proteins that are specifically their only purpose is to help um, proteins fold. And there are other proteins that hold the protein in place while it's being folded. So it's a series of actions that have to take place. But imagine one like ATP synthase, which is not just a single protein, it's a whole series of proteins, and they all have to come together, in the exact right form to be able to, and that ATP synthase just blows my mind. There's no way. It's a very, very amazing.
This is Fred Franklin, Kirby. I
Yeah,
want to say you have made an amazing presentation, and it's difficult to follow, to be honest with you.
uh,
But and I come to the same conclusion you do. How in the world can anybody see these things and see the intricacies that are in operation and not believe that there is a intelligence behind it?
I know,
It
I know.
just
I just
cannot happen by accident.
I don't, and
Yeah,
Buzz Rana, Fuzz Rana would probably criticize this presentation because what I was talking about is generally known by, it's taught in the elementary biology class,
yeah,
but I can't get past it because it's what? What is this? There's no way that could have evolved. No way.
yeah,
No way.
exactly. But the main question I have is, how in the world did they get the information needed to put the picture together?
Mm.
Yeah, and that
All of these
basically that
details,
happened.
my
That
goodness.
happened shortly after uh, the geometry of DNA was sorted out in 1953 by Watson and Crick,
Yeah,
and within Well, within 20 years after that, in 1953, within 20 years, they had all this stuff figured out. And
Wow.
how they did it, I mean, that in itself is pretty amazing.
Yeah. Probably need another 24 to 48 hour presentation to tell us how. But it is absolutely amazing.
Yeah,
There's also a theory that a lot of our diseases come from dysfunctional mitochondria,
true.
including cancer. The origin of cancer is also in the
Yes,
mitochondria.
that's very true. There are a lot of mitochondrial diseases, and that this is not just theory. This is, this has been shown apparently, and a lot of studies have been made on mitochondrial dysfunctions. And and just the very fact that we all are just we're loaded with mitochondria, and most of them work fine. The greatest majority of mitochondria work fine, but yeah, they're so complex, it's almost bound to happen that there would be some problems.
Hmm.
The second
But
second law of
but I,
thermodynamics, basically.
but I think,
uh,
Kirby, that if you look at a biology text, they will still talk about evolution. They will not talk about design,
right. Yeah.
even though design is the most obvious mechanism.
Right. Yeah. But that's not. But design is not an acceptable um, term because it's supernatural, and the only explanation for a designer is
Wikipedia will
supernatural.
call that pseudoscience.
Yeah. So this is a little bit off subject, but let me ask for inputs right now. I mentioned at the outset, and not all of you were here, I think, that I'm considering for one of our future meetings to show the newly released movie called Universe Designed. Has anybody seen that movie yet? We have. Karen and I have seen it, but it's terrific. It's basically features a lot of the RTB speakers and a lot of major theologians explaining why the universe itself uh, is designed. And so I'm thinking about showing it. It's an hour, a little over an hour and 45 minutes, or I think roughly. But I'm thinking about showing it for one of our meetings in the future, wondering if that's going to scare people away, or if more people would like to see it, or any comments.
Yeah.
Or
I, for one, would definitely like to see it.
Yeah,
okay.
I say show the movie.
Just bring popcorn.
Yeah, right. Well,
Is
and
it
then
based
I
on-
think it would be fun to discuss it afterwards.
Is it based on
Yeah.
Hugh Ross's book on Design to the Max?
I'm trying to see who puts it out, who did it.
Well, it's a special thing that was put out, but it is. I mean, it's being pushed by RTB because Hugh Ross is quoted and Fuzz Rana is quoted, and um, well, and the Steve Meyer is quoted,
Yeah,
and a lot of
I'm
the
looking here.
intelligent
They've got
design folks, and
yeah,
Sean.
Frank Turek, J. Warner Wallace, Greg Kukl,
Yep,
Sean
yep,
McDowell,
and they're all great. They're all
Mike
wonderful.
Licona.
So,
Yeah, sounds like a good cast of people.
yeah.
I'd love to see it.
Okay.
I
Well,
think you have a consensus.
One
well, if
question, Kirby.
anybody thinks it's a bad idea, I would appreciate, even if you don't want to make yourself public, if you'd send me an email or something and say,
I think it'd be a good idea. I think watch it together and then have a discussion afterwards. That'd be a great idea.
Okay. All right.
Hey, Kirby,
Good
one thing would be
idea.
nice too is if there were a way that we could, people who are interested could watch it in advance, just because probably the audio and video will be much better. Doing a movie over Zoom is work, but isn't great.
You're right. You're right. And it is. I mean, it is available. You can rent it Well, you can buy
Okay.
it for about $14,
Oh,
and
that's
that's what I
great.
would try
Okay.
to do. And you can rent it for $4. So,
Yeah.
it's not like it's going to break the bank to do it, but
Yeah.
that is for some people that might be an issue. So, we need to be sensitive about that. But
It's called Universe Design.
universe designed, yes.
Okay. Good.
Looks
And
like
and
universedesign.com.
right now the earliest that I've got that's not spoken for is June. So I'm thinking about maybe the June meeting might be that.
So Kirby, can I go back to another question on
Please do. Please
mitochondria?
do. Yeah. I didn't mean to get us off track here, but go
Yeah.
ahead.
What about the effect of fasting and autophagy on the mitochondria?
Well, I mean, you've got all those other processes that kick in when the mitochondria, when you're not feeding it with glucose. You can be feeding it with amino acids, with fatty uh, fatty acids, or with ketone bodies. Which is basically, I think that's basically a breakdown of proteins. So
I
proteins
was
is
thinking
another
I
way
was
that you
thinking about a 1 day or 2 day fast can sometimes be beneficial for the
very
mitochondria.
definitely, very definitely. Um,
After 24 hours, you tend to switch
there's
to
no,
ketones.
I've never seen anything that indicates that fasting is a problem for the mitochondria. It's just,
So
if anything, it would be exercising the mitochondria,
it's
exercising some
also
of the other
biblical.
functions besides breaking down just glucose. Because a fast, kind of by definition, should uh, stop the flow or at least cut way back the flow of glucose. Now, on the other hand, glucose, there is a certain amount of glucose that gets stored in a process called, and I can't think of what it's called, but it's actually glucose is actually stored in hundreds of glucose molecules in this storage space. But there's not that many of them that are. I mean, in the large scale of things, there are not that many of them. So you'd even burn through those in a hurry, and before long, the fasting is going to start eating into other things besides the glucose that normally goes into the mitochondria. But it's a good idea. Fasting is a good idea.
Even biblical.
Oh, you're right. I
might,
had a few other slides. I want to show one other slide here that I haven't shown you tonight. Well,
Okay.
I mean it's an
That's
eye
very
chart.
simple.
It's an eye chart, and there's no way you could read it because it's much too small. Although
Definitely evolved.
I think
Hey,
I've got
Shelley,
if I'm out.
come
Oh,
look at this.
here's part of it that's blown
Are
up.
you missing it?
Yeah, this part is blown up, and this is the big circle here is the citric acid cycle. And this down here, let's see, so that down here I think is the
No, you
this is
showed
the
me
electron transport chain down here.
all the things that are happening at the same time.
But
This is
these
just the
are
volume
all,
section.
these are all going on at the same time.
Okay, talk
And
about
there's
that.
something I counted in this chart, I counted about 250 enzymes. Well, I'm not going to hold you all here indefinitely. So let me pray for us, and then I'll stick around a while. And if you want to chat about other things, we can do that. Or these, we can do that as well, so that you won't feel guilty if you don't want to stick around. So let's pray. Gracious Heavenly Father, thank you so much for the opportunity that we've had to consider all these marvelous works that you have put together, which boggle the mind. We just can't imagine how you could have done such a marvelous thing. And the deeper I get into it, the more questions I have that don't seem to be answered to my satisfaction, at least. And I can't wait to get to heaven to find out what the true answers are. We praise you and thank you, Lord, and ask you to go with us tonight as we go out into the world. Help us to bear witness to who you are to those who don't know. And we pray for your blessing on this meeting and on all those who attended. And we pray these things in Jesus' name. Amen.
Amen.